Hello, everyone. Welcome to the OLAW webinar titled Best Practices for Conducting a Search for Alternatives and Finding Animal Model and Model Organism Information. Today is Thursday, December 9th, 2021. My name is Kate Pritchard, and I'm part of the Division of Policy and Education at OLAW. Today, it is my pleasure to welcome our speakers, Jesse Cull and Joelle Mornini. A few housekeeping details to begin. Today's webinar is being recorded. The chat is closed, but you can put any questions that you have for the speakers into the Q&A box, and we'll get through as many questions as we can at the end of the presentation. If we're not able to get to all of the questions, we'll ask the speakers to answer the questions after the webinar and then append those questions and answers to the end of the transcript, which will be posted on our webinars and podcast webpage within the next week or so. And now I'd like to introduce our speakers. Jesse Cull, MS, is the Animal Welfare Information Center, otherwise known as AWIC, coordinator at the USDA National Agricultural Library. Jessie attended Virginia Tech and pursued her Bachelor of Science degree in Animal Sciences. She then earned her Master's in Animal Behavior and Welfare at the University of Tennessee. She started her career in Iowa as an on-farm animal welfare auditor for the swine industry. In 2018, she joined the Department of Defense as their IACUC compliance coordinator overseeing their animal research labs before returning to AWIC in March of 2020 in her current role as the AWIC coordinator. Joelle Mornini, MLS, is an informationist at the NIH Library supporting the Data Services Program. She holds a master's in library science from the University of Maryland and has previously worked at a variety of biomedical libraries, including the Scientific Library at the National Cancer Institute at Frederick, the Gorgas Memorial Library at Walter Reed Army Institute of Research, and the National Library of Medicine. Welcome to you both. And with that, I'll hand the presentation over to Jesse.
Thanks, Kate, and good afternoon, everyone. My name is Jesse Cull, like Kate said, and I'm the USDA's Animal Welfare Information Center's coordinator, which is located at the National Agricultural Library. And I'm really excited to be here and talk to you guys about AWIC and how to conduct a literature search for animal use alternatives. For my portion of the presentation, I'm going to very briefly go over the Animal Welfare Act, just in terms of when and why AWIC was created. I'll quickly review the 3 R's, and then I'll talk about how to conduct a search for alternatives and end by going over some of AWIC's products and services that are available to you. So some of you might be familiar with some of the history behind the Animal Welfare Act and maybe even AWIC, but just to lay the foundation for the rest of this presentation and why AWIC was created in the first place, I wanted to briefly cover the beginning of the Animal Welfare Act. So the Animal Welfare Act, which at the time was known as the Laboratory Animal Welfare Act, was signed by President Johnson in 1966, and the real intent of this initial act was to prevent pet theft. However, it's been amended numerous times since then, but we're mainly going to focus on the 1985 amendment, which is really where the welfare of research animals came into the picture. And specifically, Senator Dole, who actually just passed away this past weekend at the age of 98, helped amend this act to state that the main thrust of the bill was to minimize pain and distress suffered by animals used for experiments and tests. And in doing so, biomedical research will gain in accuracy and humanity. So really the intent of these amendments were, again, minimization of pain and distress in research animals. And now that there are going to be so many more information requirements that were going to be asked from the IACUCs and the PIs, such as having to consider and search for alternatives to painful and distressful procedures, Congress put a provision for an Animal Welfare Information Service at NAL that shall, in cooperation with the National Library of Medicine, provide information pertinent to employee training, which could prevent unintended duplication of animal experimentation as determined by the needs of the research facility, and on improved methods of animal experimentation, which could reduce or replace animal use and minimize pain and distress to animals. And this information service later became known as the Animal Welfare Information Center in 1986. And now that we've gone over some of the history, we're actually going to transition over to the 3 R's, and we'll talk about how they came to be, how they were defined, and I'll give you some examples and why we should care about them. So the 3 R's concept started with Dr. Russell and Dr. Birch, who lived in the UK at the time, when they were asked and when they were hired by the University's Federation for Animal Welfare to look at the concerns around using animals in research. And they published their thoughts and ideas in 1959 in a book called The Principles of a Humane Experimental Technique. And they weren't necessarily against using animals in research, but instead proposed 3 areas where they felt that the research community could advance in humane techniques when using animals. And of course, those 3 areas are replacement, reduction, and refinement. Now let's get into actually defining the 3 R's and giving you some examples. So replacement is replacing or substituting living sentient animals with insentient material, which means anything that is incapable of feeling or understanding things like, let's say, algae or a sponge, for example. And the photo on the slide actually shows an example of a replacement model that is called an organ on a chip, which is a microphysiological system that's used a lot in drug testing and development. But the main example that I wanted to get in with you all is to talk about sentinel animals. And this is a newer example of replacement, and it has to do with replacing sentinel animals. And what sentinel animals are animals used as a monitoring tool to see if an infectious disease or harmful agent is present in the animal colony of interest. So it is typically done in the animal colony's bedding of interest is collected and is placed in the sentinel animal's cage to see if then the animal becomes infected with some sort of bacteria, virus, or parasite, which allows the researcher to know what disease or agents that the animal colony of interest might have. But recently, new methods such as exhaust air dust testing and PCR testing of filter media or swabs has been found to replace sentinel animals to detect those specific pathogens in the animal colony, and thus completely replace this whole group of animals that were previously required. And to go a little bit more in detail what these methods are, exhaust air dust testing uses either sticky swabs or some type of specialized filter material or media that is placed in a specific location, such as maybe at the end of a cage unit to collect as much dust as possible. And then those swabs or media are tested. And the PCR method is very similar, but they use large swabs or pieces of filter paper to test the colony's soil bedding. And some of the benefits, other than replacing animal use, is that it increases result sensitivity and accuracy and also reduces labor and cost in the majority of cases. And the second of the 3 R's is reduction. And reduction is defined as reducing the number of animals required per experiment, but still achieving robust results or statistically significant data. And one example of reduction that can be applied really in any area of research is eliminating the use of a control group of animals by allowing the main group of animals or the ones that you're going to test or treat to serve as their own control. And this not only reduces the number of animals used, but also reduces variation in the data since you are using that same animal across all groups and treatments. So for this example, if the study involved tracking the number of steps a cow has in a day, you could track this cow's A steps during a baseline period, which would then serve as the control group. And then you could track that same cow step during the treatment period, which in this case is a 24-hour line deprivation period. And that way it would reduce animal use by eliminating, again, this whole additional group of cows. And then lastly, our last R is refinement. And refinement refers to methods that minimize animal pain and suffering and therefore improve animal welfare. And again, one example of refinement that can be applied to any areas with using animals is handling and training. And again, this is important in any research or education setting because trained animals are less stressed when asked to complete a specific task and are more comfortable when they have to be handled by humans. And there's a lot of different options for training and handling opportunities depending on the species that you're working with. But for example, in mice, we use mouse tunneling rather than picking a mouse up by its tail, which we know is painful and stressful to the animal. And in rats, something that's become relatively of a hot topic is rat tickling. And that's frequently used where technicians are mimicking the natural rat play behavior. And actually I have this short video of rat tickling I'm just going to briefly show you, but keep in mind that the volume is very low on this video, so I apologize for that, but it is a really good video that demonstrates rat tickling. So let's pull it up now. And I'm just going to stop it there. Again, I apologize for the low sound quality, but hopefully the important part is that you could see how the tickling occurs and how that the rats really do enjoy it. So let me switch back to the PowerPoint. Okay. But so what? I'm giving you all this information about the 3 R's and the requirement in the Animal Welfare Act to consider alternatives if you're conducting animal research. But what's the problem? It all sounds pretty straightforward, right? Just go to the literature and search for alternatives and you've met this requirement. Well, one of the challenges with looking for alternatives in the literature is that they're extremely hard to find. And one reason this is because typically, unless the research is focused on the 3 R's or it's a main objective or topic of the study, scientists won't mention or write explicitly about the 3 R's, making these papers and information extremely hard to identify in the literature because the papers aren't being tagged or indexed on the back end as such. Additionally, some scientists may not even realize that the method or procedure they're using would be considered a 3 R's method. And again, do not explicitly state anything about the 3 R's or animal use alternatives in their paper, such as maybe using an animal as its own control. They may not realize that, and then again, write that in terms of the 3 R's in their paper. But for example, if you look on this slide at the first citation, it's about dairy calves, and it says, this is the first study to demonstrate that pair housing improves the affective aspect of calf welfare, which suggests that pair housing is a refinement method, right? Since it improves their welfare. However, you'll notice that there isn't any mention about refinement specifically in this paper. And further, if we go on to the third example, which talks about sentinel animals, it says that these media in pooled soil bedding samples is more effective than traditional sentinel methods for colony health surveillance. So this goes back to that replacement example I just went over. But by considering the context clues and interpreting the sentence as humans, we know that it's an example of a replacement method. But nowhere is replacement mentioned in this paper. And again, the reason that this is a problem is because if it's not described in these papers as a 3 R's method, then the automated indexing technology that we're using on the back end to tag or really categorize these citations, we can't just have it rely on the 3 R's terms to identify these papers, thus making it hard for individuals to find information because a lot of people searching in databases will likely use those 3 R's terms, replacement, reduction, refinement, alternatives in their actual search string to find this information. But if it's not in the paper, it's not going to come up in your results. So now that I've identified the problem, I'm actually going to take you through a real alternative search example so you can see how I construct it. So this example states that a research lab is planning multiple studies using a strain of common laboratory white mice. They're planning to house the mice in stacked wire cages with feeders and waterers. Mice are individually housed in steel cages with no bedding or other enrichments. So our question is, what 3 R's modifications could be made to this housing model to improve animal well-being and reduce stress? So the first thing we want to do is identify the different topic areas within the question to then form those different parenthetic statements or concept groups that will then create our full search string. So for example, for this search and for the question that we want to address, we want to make sure we have a concept on housing terms, animal keywords like mice, and then 3 R's alternatives terms that are related to their housing or environment, such as maybe environmental enrichment. We don't necessarily just want to use the term refinement or the 3 R's. And again, the reason I picked these 3 topic areas is because I'm looking for ways to improve the animal's environment. So I need to include words relating to housing. I need to include terms related to mouse, and then of course, housing alternatives. And I also can't just use these one terms for each concept because there's going to be a ton of different ways to refer to housing or let's say the animal mice, such as mouse or also murine. And if I don't include all other synonymous terms, I'm likely going to miss a lot of valuable information because not all scientists write using the same terms to refer to the same thing. So I hope that makes sense. We'll go through more synonymous terms in a second. And so the next few slides I'm showing you are just some 3 R's terms that we came up with that are examples of each R that you can include in your search string and likely lead to more productive results than using replacement, reduction, or refinement. So for example, this slide is showing you refinement examples or related terms, and specifically the ones I've highlighted in yellow could actually even be used as keywords in this search example that I'm going over on environmental enrichment. And I'm not going to, again, go through all these. They're mainly here for your reference, but this is an example of keywords on reduction that you could use. And then lastly, here are some replacement keywords. And again, my main point in showing you this is that using these terms in your search string is going to lead to more productive search results than just adding in the terms 3Rs or replacement, reduction, refinement into your search string. Because like I mentioned before, a lot of scientists won't include those terms in their papers. And if they do, it likely isn't in the context of the 3Rs, which is why you might not be finding any relevant results. But back to the search example, here again are the three concepts that I just talked about, but written out on the slide with various synonymous terms, and then the Boolean operator or in between each term. So for housing, I included other key terms like cage or enclosure. For animal, I included mice, mouse, the scientific and Latin terms, murine and mus. And then lastly, for my 3Rs terms, I included social housing to see if mice can be housed in groups. And I also included specific enrichments like toys or bedding or nesting material. So again, any terms related to housing opportunities that can improve their welfare. And I'm not including, and I'm making sure to include multiple terms that refer to the same thing, again, so I can capture all relevant information. For example, if I only included the term environmental enrichment, I'm not going to get any papers that talk solely about social housing or maybe specifically about nesting material for mice. And it would only bring back papers that specifically talk about environmental enrichment. And the same applies for housing. If I'm only using the term housing, I'm going to miss papers that refer to housing maybe as solely as cage or enclosure. So I hope that makes more sense now. And one other thing I wanted to mention before moving on is that you'll notice I didn't put quotation marks around the term bedding. And that's because if it's a single word and not a phrase like animal welfare or social housing, you don't need quotation marks. However, they don't hurt to add if you do want to add them. Just wanted to mention that. And then some of you may have also noticed the asterisk on the term nesting material. And what I'm doing there is truncating the word material so that it captures all word endings of that term, such as nesting material or nesting materials. Another term we typically like to truncate is the term behavior. So instead, you would put an asterisk after the V in behavior. So you would get terms like behave, behaves, the European spelling of behavior, behaved, etc. And then lastly, you would take your three search strings and join or connect them with the Boolean operator and, which tells the database that you want to search for papers that have at least one word from each search string in the papers. And now I'm going to go ahead and show you a very quick live demo in PubMed. So let me transfer over. So this is the advanced search screen of PubMed. And the first thing I want you to draw your attention to is that this all fields box. The first thing that I always do is change it to title and abstract, because I think all fields is just a bit too general and broad, and it's going to usually bring back way too many results. So I'm going to change the title and abstract. I'm going to put in my first search string on housing keywords. I'm going to say add. It's going to add it to the query box, and you can see it's just searching each term by title and abstract. I'm then going to do my search string on mice, and then now it switched it to and. Remember I talked about the Boolean operator and. So it added that down here, and you can see it's separated by the Boolean operator and. And then I'm going to add my last search string on 3Rs alternatives. Same thing. Here's the full search string. I'm going to press search. And here are all my results. And you'll notice that it does search over 600 results, which is likely too many for anyone to go through. But this is where modifying your search string comes into place. So you can get a more manageable amount of citations and a set that is hopefully more targeted as well. And one thing you can do to modify it is add an additional search string at the end. So a fourth search string separated by the Boolean operator and, which would result in less citations since you're putting more parameters on that search or making it more specific. And just for example, that fourth search string could be on keywords related to your area of research, such as if you're in the area of toxicology or immunology or nutrition. or whatever your area is. And this is going to reduce the amount of citations you get. Another easy way to reduce the number of citations is to search by only the title field instead of the title and abstract field, which is going to lead to a lot more relevant results because those keywords are now required in just the title field. So let's try that. And again, this is a trial and error process. If this came up with 0 results, we would know that title is too specific and we'd have to figure out another way to modify it. Okay, you might have to go back to the all fields. Okay, so now I have all my search strings. I have environmental, housing. Okay. And now I'm going to go ahead and search that. And you'll notice this brings down the results to only 34. So it's significantly reduced and a lot more manageable for you to go through. And I don't have time to go through them now, but I did want to show you how I quickly refined it. So I'm going to go back to the PowerPoint. Okay, so once you get your set of citations to review, you'll want to skim your results to see how relevant they are. If most of them are relevant, then there's no need to refine it further. However, more than likely, if it's your first attempt at searching, you will need to refine your search string. And another way to refine your search results might be to limit by publication years. So limiting your results to only things published in the last 5 years or maybe 10 years. Or like I mentioned before, limiting a title or title abstract. Keep in mind that what I'm showing you is a very basic search. So there's a lot of room for improvement. And also note that it really, again, is a trial and error process. It's highly unlikely that you're going to get the perfect set of results using the first search string you created. And then once you've refined your results to where you want them, the last step is to save them in a citation manager like EndNote, RefWorks, or Zotero. And what these are essentially a citation database that helps you save, organize, and share your research citations. And I did just want to highlight one of those citations that were in the set of 34 results I just showed you, just so you know that this whole search string and process I'm taking you through actually worked. And this paper specifically talks about how using nesting material and shelters improves their welfare without interfering with the experimental outcomes. So again, a study that directly answers a question we sought out to address. And then lastly, before we close out this section, these are some items I put together that would maybe be considered red flags when conducting a literature search. So for example, if you're only searching one database, you're going to miss a lot of information because one database never covers a topic 100% comprehensively. Another thing, if you're doing the search last minute, then you're realistically less likely to incorporate alternatives into your research, which again is why the consideration of alternatives should come before your protocol is written, such as during the grant writing phase, because if you already have the funding, it's highly unlikely that you're going to make any 3Rs modifications at that point. If you only put in terms related to painful procedures, it's not going to be very helpful in finding alternative methods because you're mainly going to get results with that procedure in which you're trying to replace. Unless you know it's a painful procedure that like oral gavage, where a lot of alternative research has been done. And also if you use the term alternatives in your search string, you're likely not going to find anything useful unless you're working on a teaching or toxicology protocol, which does use that term in the context of the 3Rs more frequently than other animal research areas. And then lastly, restricting your search by years can, again, be helpful to refine your results, but keep in mind it's also going to greatly limit the citations you're going to get back, so you may miss something. And then just briefly, this is a list of databases that we typically use and recommend when you're looking for animal science or animal welfare information. And more specifically, Agricola, PubMed, Web of Science, and Scopus are great if you're looking for animal use alternative information. And one thing to keep in mind when using Agricola is that, yes, you can search it freely through the platform on NAL's website, but we recommend that you search Agricola through your own institution in a multi-database system like EBSCO or Ovid, if you have access to it, because using the NAL's platform of Agricola is sometimes a little hard or clunky to use on its own. And now we're going to discuss some of the resources that NAL and AWIC makes publicly available to you. The first I wanted to go over was the AWIC website, which some of you may or may not already be aware of. But you can find us by going directly to the link on the slide or from the NAL homepage. And you can find all kinds of peer-reviewed information on animal welfare, as well as other topics like what's in the news, updates on laws and regulations to animal welfare, bibliographies on hot topics in animal research, and all of our upcoming training opportunities. And speaking of training opportunities, AWIC offers a free day-and-a-half in-person workshop at NAL on meeting the requirements of the Animal Welfare Act. However, the past year or two, we have had to modify the workshop to fit a virtual setting, but we're hoping that sometime in 2022, hopefully October, we'll be back to in-person. But you'll notice that our next workshop is virtual on March 9th. So make sure to register if you're interested. And within this workshop, we do dive deeper into the Animal Welfare Act and animal welfare in the legislation. We discuss the 3 Rs in more detail. We also talk about various databases and other resources and organizations that are helpful for finding information on the 3 Rs. And then lastly, one of our main takeaways from our workshop is an improved understanding on how to conduct a literature search. So yes, we focus on alternatives, but the concepts that we teach can also be applied to any type of literature searching. And then other than our in-person workshop and live webinars, we also now host our workshop online in a pre-recorded, self-paced, and free format. And while it doesn't have the in-person activities and interaction that our other workshops and webinars do, if you can't make one of those trainings and you still want access to the information, this is one way to get it. And then kind of like we're doing for this presentation, we do offer free shortened or condensed webinars on request. The difference between these and our workshops is that we can really tailor the topics of the presentation to your institution's wants and needs, meaning we can really hone in on a specific topic, and the length of the training can vary depending on what you're interested in. And then AWIC does offer reference and alternative literature searching assistance upon request. So if you're having trouble finding alternatives for a specific animal-related procedure or method or model, feel free to reach out to AWIC for help. We just developed and posted a literature search request form on our website that makes requesting a literature search from us a lot easier. And then also just in general, if you have any animal-related questions, not just related to scientific literature or alternatives, feel free to reach out to us. And if we can't find that exact information or answer, we can at least point you in the direction of someone who does. And then lastly, if you're interested or not already signed up, AWIC does send out a monthly newsletter, which covers topics such as information on the 3 Rs, highlights related to AWIC, the Animal Welfare Act, tips and tricks on literature searching, and any upcoming 3 Rs related conferences and trainings, among other information. You can just click on the link on the slide and subscribe. And again, we only send this out once a month via email, so don't worry about being bombarded with tons of emails. It's just a good way to stay up to date on 3 Rs information. And this is our contact information if you ever need to reach us. And with that, I will go ahead and hand it over to Joelle.
Great, thank you, Jessie. Hi, everyone. My name is Joelle Mornini, and I'm an informationist at the NIH Library. Today, I'll be providing an overview of best practices for finding animal model and model organism information. We'll take a look at how to locate biomedical research articles, patents, NIH-funded research projects, and genetic information related to animal models and model organisms. After completing this training, you'll be able to describe the difference between animal models, research organisms, and model organisms, and how these concepts are related. You'll be able to identify requirements and resources for the NIH model organism sharing policy. We'll take a look at how to locate biomedical articles related to animal models through PubMed, and how to locate patents related to animal models on a SPOSnet. You'll be able to locate NIH-funded research projects that utilize specific research organisms through NIH Reporter, and also identify publications, patents, and clinical trials related to these research projects. We'll look at how to identify genetic information and biomedical literature about specific research organisms through the taxonomy browser from the National Center for Biotechnology Information. And finally, we'll also look at where to find model organism databases, which provide genetic and biological data about specific model organisms. First, let's look at the definition of animal models, model organisms, and research organisms. According to the National Cancer Institute's Dictionary of Cancer Terms, an animal model is an animal with a disease either the same as or similar to a disease in humans, and the animal may be used to study the development and progression of diseases and to test new treatments before they are given to humans. Animals with transplanted human tissues, such as cancer, are called xenograft models. Many animal models may be genetically modified based on the requirements of the study. The article, A Brief History of Animal Modeling, published in 2013 in Missouri Medicine by Erickson et al., describes how the first transgenic mouse, where the mouse carries additional genetic materials from an unrelated organism, was developed in 1976, while the first knockout mouse, where genetic materials is inactivated, was developed in 1987. Animal models are often used in preclinical research before testing new treatments on humans during clinical research. The National Institute of General Medical Sciences broadly defines a research organism as any creature that scientists use to study life, which can range from single-cell bacteria to more complex organisms like mice. A research organism is identified as a model organism when it is used to examine elements of human disease. Reasons an organism may be used as a model organism include biology that is similar to humans, or a lot of information may already be available about the organism's genetic makeup. Some examples of model organisms include fruit flies, mice, rats, and zebrafish. The term animal models and model organisms can often be used interchangeably. In this training, we will take a look at terms related to animal models within controlled vocabulary and classification systems to more effectively locate journal articles and patents related to animal models. We will also take a quick look at resources and research strategies to locate NIH-funded projects and genetic information related to research organisms and model organisms. Just to summarize, model organisms like mice, rats, fruit flies, and zebrafish are often used as animal models in the study of human disease. Model organisms and animal models may be genetically modified based on the needs of the study. The terms animal models and model organisms can often be used interchangeably. Now we're going to take a quick look at resources for learning more about the NIH model organism sharing policy. The NIH policy on sharing of model organisms for biomedical research is an extension of the NIH policy on sharing research resources, and it went into effect on October 1st, 2004 for applications or contract proposals to NIH. This policy applies to extramural investigators funded by NIH grants, cooperative agreements, and contracts. A description of the policy is in a brochure available at grants.nih.gov and states that all NIH applications and proposals that will produce new genetically modified variants of model organisms and related resources are expected to include a sharing plan or to state why such sharing is restricted or not possible. You can check the NIH model organism information page to find links to the original NIH guide notice related to the policy, an FAQ on sharing of model organisms and related resources that includes a searchable list of commonly asked questions, links to examples of plans for sharing of model organisms and related resources, including a simple plan, complex plan, and plan for sharing mice, and a brochure about the policy in PDF format. Next, we'll take a look at how to find biomedical research articles related to animal models using PubMed. PubMed is a free biomedical literature database from the National Library of Medicine, and it covers over 33 million citations for biomedical literature for Medline, life science journals, and online books. Medline is the main bibliographic database from the National Library of Medicine, and it contains over 28 million references to journal articles in the life sciences and biomedical fields. Journal articles included in Medline have medical subject headings, which are also called mesh terms, added to the article records to make them easier to find with consistent and controlled vocabulary. In PubMed, you can try searching for articles with models animal added as a mesh term. All narrower mesh terms under a broader term are also automatically included in the search. So search for articles with mesh terms models animal will also automatically include search for articles with mesh terms like disease models animal. You can visit the mesh browser at ncbi.nlm.nih.gov/mesh to look up mesh terms and then add them to PubMed. So let's visit the mesh browser. And in this search, I've searched animal models. And we come up with just one related mesh term models animal. And you can see there's a description of the mesh term followed by subheadings that can be added to further narrow with the search. There's a number of entry terms, which are various synonyms related to the term, in this case, animal model. And if we scroll down, you can see all the narrower terms under animal model, which includes disease models animal, and then a number of narrower terms related to various diseases. And if we want to search this term in PubMed, you can just choose Add to Search Builder and then search PubMed. And you can see there's around 611,000 publications that have been tagged with the mesh term models animal, or one of the narrower terms. So this screenshot shows a search in PubMed for articles labeled with the mesh terms models animal, and an additional search term to narrow the search to only articles related to melanoma. Note that the second search term doesn't include any search field tags that would limit the search for the term only to a specific field, like title, abstract, or mesh terms. If no search field tag is added to a PubMed search term, then PubMed automatically applies a feature called automatic term mapping to the search term. Automatic term mapping can locate various synonyms, mesh terms, and singular or plural versions of the search term in order to expand the search. You can go under the Advanced Search Builder on PubMed to view the History and Search Details table, and choose to expand the Details column to view how your search term was expanded using automatic term mapping. So if we go back to our search, we can type in "and melanoma" in order to narrow our search further, just related to a specific disease. And if we go under the Advanced Search, we can scroll down and see how our search actually mapped using the Details column. And you can see that since we specifically added the mesh field tag, it searched just across mesh terms for models animal. But for melanoma, since there wasn't any field tags added, it searched across a mesh term that was automatically linked to melanoma, and it also searched across a variety of plural, so melanoma, melanomas, melanomas, across all fields. So the automatic term mapping is a great tool for expanding your search, but I always recommend double checking under the detail section just to make sure that it's actually mapped to correct mesh terms and related synonyms. Now we're going to take a look at how to locate patent documents related to animal models through the free patent database ESPASnet. ESPASnet is a free patent search tool from the European Patent Office that searches across more than 130 million patent documents. You can try using the Cooperative Patent Classification, or CPC browser, to find classifications related to animal models and model organisms. For example, if you search by the term animal model within the CPC browser, you can find relevant classifications like A01K2267/03, animal model, e.g. for tests or diseases. You can then choose the checkbox beside a relevant classification, which will automatically select all subclasses, and then choose Find Patents in order to search for all patents labeled with this classification or related subclasses in ESPASnet. So I'm going to go ahead and go to ESPASnet. And you can see we can choose to search within the classification search, and I'm going to search for animal model. And then you may need to expand various terms in order to view more relevant subclasses. In this case, we come across animal model, e.g. for for tests or diseases. And you can tell if there are narrower terms beneath it by the number of dots beside it. More dots equals a narrower subclass. And if you see \L low, that just means it's going to search across all of the subclasses as well. So we'll click on Find Patents. And that brings up about 6,900 patents related to that subclass or related to that classification. You can also try using the advanced search on ESPASnet to search for keywords in the title, abstract, and claims of the patent documents, like organism name or disease, and combine with CPC code searches. You can combine searching by classification and keyword to find patents related to animal models for specific organisms and diseases. For example, this screenshot shows a search using the animal models, e.g. for test or disease CPC code, in combination with a search for keywords mouse or mice and hepatitis in the title and abstract of the documents in order to locate patent documents related to hepatitis in mouse models. So just going back to our search, I'm going to turn on the advanced search. And then we can add additional fields to narrow the search further. So I'm going to just choose to search within a text field, title or abstract. I'm going to choose any so that it'll include a Boolean operator or between each of the different searches, each of the different search terms. And then I'm going to enter mouse or mice. I'm going to add one more field, and I'm going to enter hepatitis. And I will run the search, and that gets us down to about 25 search results. And if we want to view one of the actual results, you can click on the result, and that will open it in a separate window. You can view the IPC codes, the International Patent Classification, which is actually an older classification system, which the CPC is built upon. You can scroll down and view the title and abstract. You can use Patent Translate if it's a non-English language patent in order to translate it using machine learning into whatever language, usually English. So this is one tool that's helpful for translating non-English patent documents if you need to just get a quick idea of what they're about. You can view the different sections of the patent. And if you click on original document, you can actually download the full patent document just by going under download and then original document. You can also view the patent family, which is filings of the patent around the world. If you click on IMPADOC family, that's usually the broader patent family that will include many filings of related technologies related to that patent. And if you want to download the entire list of patent documents, you can click on any of the checkboxes and then click download and list either into Excel, or you can even download the front pages of the documents. So just now I'm going to launch a quick poll. It looks, let's see. All right, so what database tools can be used to find more targeted results related to animal models? And just select all that apply. And the options are mesh terms in PubMed for biomedical articles, automatic term mapping in PubMed for biomedical articles, basic search in PubMed for biomedical articles, CPC and ASPOSNet. Let's see.
I think I had to relaunch it, so it should be open now.
Okay, thank you. I'll let it run for about 30 seconds. All right, give it about 10 more seconds and we'll go ahead and see what the answers are. All right, so I'm going to go ahead and show the responses. And so the correct responses are medical subject headings and the cooperative patent classification. Those are two ways to narrow your search. If you want to expand your search, especially in PubMed, you can use automatic term mapping, and that can be a helpful tool for expanding your search. So moving on. Now we're going to take a look at how to locate NIH-funded research projects related to specific research organisms using NIH Reporter. You can use NIH Reporter to find NIH-funded research projects, investigators, publications, and patents related to specific research organisms. Here are some tips for locating projects that utilize specific research organisms. Under the Advanced Project Search, choose Advanced Text Search, and then enter all versions of the organism name, both the common and scientific as well as plural, using the Boolean operator or between each organism name search term. And you can also use quotes around phrases and parentheses to group all versions of the organism name. And then also use and, the Boolean operator and, to add additional search concepts. So just as an example, to locate projects related to cancer and fruit flies, you could use fruit fly or fruit flies or Drosophila melanogaster or D. melanogaster and cancer within your search. For a shorter list of relevant projects, try limiting text search to project title, project terms, and project abstracts. So let's go ahead and go under NIH Reporter. And we'll click on Look for Additional Fields, and this will expand the full search. or the full menu. And I'm going to just copy the search strategy that was listed in the slide. I'm going to paste it in the search box and go under advanced. I've made sure that project title, project terms, and project abstracts are selected. And then I'm going to just choose the fiscal years that I'd like to search. I'll just search for within the last few years, and I'll run the search. And we get our search results. So just looking at the search result box, under the search results, you can view NIH-funded projects where any version of the organism name is listed in the project title, project abstract, or terms, and also choose to view lists of publications, patents, and clinical trials related to those projects. You can find links to the publications on PubMed, links to the patents in the U.S. Patent and Trademark Office database, and links to clinical trials at clinicaltrials.gov. So just going back to our results, if you click on one of the project results, you can view a description of the projects with highlights of the search terms, so related to your research organism and other research terms you've added. And then you can find related publications, patents, clinical trials, and more. If you click on the specific tabs, you can also view lists of publications, patents, and clinical studies related to these particular projects. And you can see that the publications will usually link to PubMed or Google Scholar, patents will link to the USPTO database, and clinical studies will link to clinicaltrials.gov. Now we'll take a look at how to find genetic information for a specific research organism through the taxonomy browser from the National Center for Biotechnology Information. We'll also look at model organism databases where you can locate genetic information related to specific model organisms. The taxonomy browser is a tool from the National Center for Biotechnology Information, or NCBI, which is part of the National Library of Medicine. You can use the taxonomy browser to find links to genetic information and biomedical literature related to specific research organisms. Users can search the taxonomy browser by the common name for the organism, like zebrafish, or by the scientific name, like Danio. So if we go to the the taxonomy browser page, it's maybe just go directly since it seems like it's freezing up a little bit. One thing I recommend is click on Browser, and this will allow you to also just view a pre-made list of research organisms. You can also always just try searching by research organism. You can search by either the Latin, scientific, or the common name. Seems like there might be some issues with the website, but let's search for zebrafish. And you can see that it'll bring up the Latin name, but you'll also see some information related to the common name beneath. And when viewing it, you'll view a list of, it says the entrance records. This is just a table where you can find links to various NCBI databases with pre-made searches related to that specific organism. And you can also scroll down and find links to external resources related to that specific research organism, including model organism databases like the zebrafish model organism database, which we'll discuss in just a second. So pages for specific organisms on the taxonomy browser include direct links to research results related to that organism and their genetic makeup within other NCBI databases, such as the nucleotide database, which includes genome, gene, and transcript sequence data, the protein database, which includes protein sequences, the genome database, which hosts information on various entire organism genomes, including sequences, maps, chromosomes, assemblies, and annotations, and PubMed Central, which includes full-text biomedical articles. Pages for specific organisms on the taxonomy browser also list links to external information resources related to the organism, such as model organism databases. Model organism databases, also known as MODs, provide in-depth biological data for commonly used model organisms. These databases are often funded by grants from various government institutions, including NIH, and may be maintained by government, academic, or other nonprofit institutions. Examples of MODs include FlyBase, which covers fly genes and genomes, Mouse Genome Informatics, Rat Genome Database, and the Zebrafish Information Network. Each model organism database may have varying layouts, tools, and data available, depending on the user needs and the resources available to the database developers. A toolkit created with NIH funding is available at gmod.org called the Generic Model Organism Database Project, which offers guidance on open source software tools for managing, visualizing, storing, and disseminating genetic and genomic data. To locate model organism databases for specific research organisms, you can try checking the External Information Resources section for organism pages on the taxonomy browser. Just for example, the zebrafish page on the taxonomy browser links to ZFIN under the External Information Resources section. You can also try the Online Bioinformatics Resources Collection, or OBRC, by the Health Sciences Library System at the University of Pittsburgh. Some links may be outdated on the site, but it's still a useful portal for trying to locate MODs for specific organisms. You can browse databases for vertebrate and non-vertebrate organisms. So just as a quick summary of what we covered today, model organisms are often used as animal models in the study of human diseases, and the term animal model and model organism can often be used interchangeably. You should be aware of the NIH model organism sharing policy, and the policy applies to extramural investigators whose research will be generating new genetically modified model organisms. And you can find information on animal models and model organisms through PubMed for biomedical articles, SPOSNet for patent information, NIH Reporter for NIH-funded research projects, Taxonomy Browser, which can link to genetic information, as well as biomedical literature through PubMed Central, and model organism databases, where you can find a variety of genetic information. And here's my contact information. And I believe I'll pass it back over to Kate.
Yes, thank you so much. That was great. Thank you, Jessie and Joelle. We do have time for one or two questions. And I see that we do have a lot of questions that are very specific to certain situations. And so I would encourage the participants to contact the NIH library or AWIC to get answers to those questions. So I'm going to ask one very general question. Do you have any tips for iCooks that evaluate alternative searches associated with animal use protocols? And Joelle or Jessie, you're both welcome to answer this.
Yeah, I can take a stab at it first. Yes, I meant to actually include a slide on that, but some tips that I think the iCook should keep in mind is if when you're reviewing a literature search, look at their keywords. One of the biggest mistakes we see is that there's only one keyword usually added, and might be like heart and or cardiac and canine and cancer, or something like that model, where you're not adding all these additional terms, which is really going to strengthen your search. And then if they could say they come up with 0 results, usually a red flag. I mean, there might be in some situations 0 results with a perfect search string, but it's kind of unlikely. And then keep in mind limited publication years. We typically like to go back at least the last 10 years, but sometimes 5 is sufficient depending on the area of research. And always having a librarian on staff of your iCook committee is really helpful, because obviously not everyone on the iCook can be experts on literature searching, but the librarian serves as that expert and can better evaluate the search.
Thank you. Joelle?
Great, thank you, Jess. I definitely agree with Jessie. It's always a good idea to have a librarian either working with the iCook, or to send any requests to them if they need assistance with literature searches, especially when doing it through PubMed. And oftentimes you may not be aware of some of the other databases that may be accessible through your institution's libraries, such as Embase or some other databases where you may be able to expand your literature search and find additional resources.
Yeah, and then also, again, red flags if they're just using the terms like alternatives and they say there's 0 results. You really got to think outside the box, like those 3 slides I showed with examples of the 3 R's. Those are going to be so much more helpful, like using the terms in vitro or mannequin or imaging or like real examples. That's going to really strengthen your search. Yeah, sorry, Kate.
Great, thank you. Do you want to flip to the next slide, Joelle? All right, so we have run out of time, but thank you both again for that in-depth discussion, and we really appreciate your time, effort, and experience. Our next webinar is going to be next spring, and the topic is yet to be determined, but we'll make sure to advertise that in advance. So until then, stay safe and healthy, and thank you and goodbye.
Thanks, and yeah, reach out if anyone has any questions.
Great, thank you so much.